scientific paper. Because I guarantee you, if you haven't read one before, you're reading one now. If you haven't presented one before, you will be at some time in the near future. So you guys good on your posters? You know the deadline is midnight tomorrow night. Upload it as PDF, one person from each group. I think I said. So the last person was the person that uploaded the abstract. So let's make it the same person to upload the poster, if that's all right. I just don't want to get it too confusing. So if you uploaded the abstract, your job is also going to be to upload the poster, okay? And if you're the first person on the list and you've got everybody together, obviously your job is to kind of coordinate everyone and make sure that everyone submits their part on time. All right. So, but before we're going to do any of that, we are going to do a little review from last time. On Friday, we talked about plant communication. And I talked about signal transduction pathways. Remember, we talked about transduction, carry across. Signal transduction pathways carry information from the outside of the cell to the inside of the cell. And they cause changes, they make things happen. So, draw like a generalized signal transduction pathway. Make sure you label all the important elements. There are only 4 important elements in this pathway. So take a couple minutes to write it. Try to write it in your notes. So I put up this version on Moodle as well, so if you want to take your notes on the kind of white background, it's also uploaded to Moodle. I just did it, so if you looked at it, if you downloaded it before, say, 9:30 this morning, it is up now. So however you prefer to write on. Take a few moments to finish up. generalized signal transduction pathway, okay? It's nothing really specific. So you've got the cell membrane here, right? You may or may not have a cell wall, right? Plants have cell walls, animals don't. So basically the information has to get from the outside of the cell to the inside of the cell. And the cell has to do something about it, right? So we've got the three phases: reception, where the information is picked up by the cell; transduction, where it's carried across and into the cell cytoplasm; and response. So the four main components, right? The signal, the first one. It could be a hormone, it could be any sort of protein that's going around the cellular environment. Or remember we talked about the Greening response in plants, it could even be an environmental stimulus like light. The signal binds to a receptor. This receptor is located within the cell membrane. And so it causes a change in the receptor such that on the inside of the cell, second messenger molecules are produced. These are small, diffusible molecules that spread through the cytoplasm very quickly. They can be things like cyclic GMP. There's also an equivalent cyclic AMP, which is very common, or calcium ions, for example. Could be a lot of small, diffusible molecules. Now, I have a question for you. So calcium and cyclic GMP are called second messengers. Where's the first messenger? If these guys in the cytoplasm are the second messenger, what would you call the first messenger?
Would it be like one of the hormones, kind of like the signal?
Right, yeah. The first messenger would be the signal, whatever that is. It's not a term commonly used, but it's how the second messengers got their name. So yeah. So the signal is the first messenger, the receptor receives it, produces second messengers, which diffuse through the cell, and they activate a response. And that can be changes in cell physiology, which can be short-term, right? So only a few minutes to a few hours. Or they can be long-term. Genes can be turned on and off, and some of these changes last for the lifetime of the cell. Okay, so any questions about signal transduction? We know the four main components. The point is to carry information from outside the cell to in. Good? All right, good. So we talked about Darwin's other experiment. Charles Darwin experimented on other things besides the theory of evolution. And one thing he experimented on was phototropism in plants. So show me what all the things that Darwin did. Don't worry about what the other people did, right? Including all the controls that he did, right? So he controlled for different factors when he was doing his experiment. That's a really important part of the scientific method. And then tell me, what was his final conclusion? questions to finish up? So, let's show what happened, right? So, Bill Charles Darwin was a good Englishman, had a little greenhouse, and he was interested in phototropism in plants, the bending that plants do towards the light. And so he had hypothesized that only part of the plant is responsible for this movement, and he hypothesized that it was the tip of the plant. So the first thing he did was just lop off the tip, okay? And when light was shown in the plant, it did not show the phototropic response. Right? But remember, it's too simple just to say, okay, case closed, it's right. Right? Because lopping off the tip of the plant is kind of a drastic thing. So he's like, okay, maybe I just damaged the plant so much that it couldn't respond, not that it wouldn't. So he covered the tip with an opaque cap, right, which blocked the light from the tip. And that also failed to show the phototropic response. But again, he was concerned maybe that the cap or the weight of the cap or the fact that it's just a very artificial situation affected the plant's phototropic response. So he replicated the cap, except he made it transparent to light. And there, he got the phototropic response. He also tried to cover the lower part of the plant to see if he could block the movement, but it didn't block the movement of the plant. But these three are really the primary ones that show, right? It was the tip, and it wasn't the cap, it was the fact the cap blocked the light. Brooke?
So when the light is blocked by the cap, is that when it starts moving? Or is it when the plant is exposed to the light, the plant moves to the light?
Right, so remember from the little video I showed on Friday, when the corn shoots are growing, you turn the light on, they bend their tip towards the light. That's the phototropic response.
Mm-hmm.
So what Darwin did was try to figure out what part of the plant was responsible for that. And so he said he hypothesized it was the tip, and when he removed the tip, there was no response, right? No bending. When he covered it with an opaque cap so no light could get to the tip, there was also no bending. But when he covered it with a transparent cap, so light could get to the tip, there was bending. So he said, okay, it must be in the tip, and it must be because of light. Okay? That's pretty much all he could really conclude from this, right? We talked about how experiments go on, and as technology and things improve, people are able to ask kind of more finer and finer questions about things. But he did a pretty good job for the middle of the 19th century. So that was Darwin's other experiment. Any questions or anything about plant communication we talked about on Friday? Okay. So I'm going to talk about the Silvergeld paper, and that is on Moodle. I want you guys to go, if you don't have it already, go to Moodle, download it, and open it up on your iPad just for a moment. So if you look through this is a fairly standard formatted scientific paper. So you've got the title and the author, or authors, right? At the top, you have the abstract. So generally, an abstract is a brief summary of the main findings of the paper. In general, the abstract is available to you free on databases. I'm not sure if I mentioned this to you. So generally, scientific papers that are less than a year old are called embargoed. You have to pay for access. The library subscribes to some journals, but sometimes the cost for accessing a journal is $10,000. We can't afford that. So, but after a year, all papers become free for everybody. It's part of the specifications when you accept a grant from the National Institute of Health or the National Science Foundation. You agree to that. Your papers can become free after a year. But the abstract is almost always free, even if the paper is still embargoed. Okay. Then the first part of the paper is going to be the introduction. An overview of the topic for a general audience, but it is pitched for scientists. It's not really pitched for non-scientists. So the introduction usually goes for a few paragraphs. In this case, it goes a little bit longer. Generally, sort of somewhere in the last paragraph is the author's hypothesis. So when you get to the end, the last paragraph of the introduction, they'll say something like, therefore, we believe that the tip of a corn shoot is responsible for phototropism, and we're going to test that by covering it up, kind of thing. So usually you get to the hypothesis kind of right when they're transitioning into the method section. Methods is very specifically how we did this. So you put chemical reagents, equipment, animals, how you did the statistical analysis, you put them all in the method. And you say, we got this reagent from this company, we use this statistical package on this computer platform. It's very specific, or it should be anyway. The idea is that a methods section should, in theory, tell anybody who wants to replicate this paper exactly how to do it without asking anybody any questions. So, in and of itself. Now, Silbergeld doesn't have a method section because Silbergeld is what's called a review paper. So does anyone know what a review paper is, or what it's kind of opposed to in the scientific literature? There are review papers, which are a small fraction of the scientific literature, maybe 5–10%. And then the other parts are what are called primary scientific articles. So primary scientific papers are the ones that are showing something new. Hey, we just discovered this. So a couple of ways to determine which type of paper it is. Now, oftentimes, reviews will be labeled as such. In the database, it will say review, but sometimes it doesn't, or sometimes it's harder to figure out. Sometimes the title says a review, but again, not always. So the primary scientific paper, a couple of little tricks is the authors are the ones who do the experiment. The title is usually very specific. So, right, toward the 21st century, lessons from lead and lessons yet to learn. Kind of general, right? That's more common for a review paper. A primary paper would be something like lead absorption in human T125K cells under exposure of light for 25 seconds or something. Very specific. It's about a very specific experiment. And it has a method section where they tell you how they did their experiment. Now, a review is the authors did not do the experiment. So Silbergeld did not do all the experiments she's talking about here. It's a very general title. has no method section. So a review is basically a summary of a subsection of the scientific literature that somebody puts together to basically help busy scientists get a handle on an area of study that they may not be entirely familiar with, right? So the primary literature, tens of thousands of primary articles come out every year, more than any one human being can keep track of. And so review articles are designed so that someone can take a bunch of primary scientific papers, kind of weave them together into a single story, and then present them to a scientist who may not be completely familiar with the story, but wants to learn more, or is trying to get familiar with the area, but doesn't have the time to pick a whole bunch of primary papers. Usually the author of the review is an expert or in the area and knows, I'm going to pick these papers because these are the most important ones. So a review paper, like Silvergale, doesn't have a method section, but primary scientific papers do. The next part is the results from experiments, and that includes most of the figures in a paper. So, right, we've got drawings, we've got tables, we've got graphs. These are all results. In the case of a review, these are obviously results from other people's experiments. Then you have the discussion conclusion at the end. It isn't really clearly labeled here in the Silvergale paper, but it's kind of on the second to last page. So the implications of the findings, possible caveats. You guys know what a caveat is? Caveat emptor, the buyer beware. So caveats are things that the reader should be careful of, right? So maybe the things aren't quite as simple as I made them out to be, and here's a complication. So kind of letting people know that, this may not explain the entire story. And sometimes people create models to kind of explain how they think things are working. It's a good way to kind of convey how you think some mechanism is working, but they may not be accurate. Usually they're not. And then the references at the back. This is the literature, the previous work done by other scientists that this author used in this paper. So they're usually back, they're usually numbered. And this is a really good place to go. So if you're interested in lead toxicity and you're reading a review by somebody who's an expert in the field, you know that the primary articles they chose were probably pretty important. So this is a good place to pick some articles to start with. Okay, so that's how you read a scientific paper. You start with the abstract, introduction, methods, results, discussion, references. Right? Now obviously, right, there are some parts that are more important than others, especially if you're a student presenting a paper, right? The introduction is important because it's going to get you familiar with the topic. The results are really the heart of the paper, right? This is what we found. If you're ever in a situation where you have to read a paper for class and it's 10 minutes before class and you haven't read the paper, the best thing to do is read all the figures. So most figures are printed with a figure legend, which kind of tells you a little bit about the figure. But if you read all the figures, they should, not always, but they should, in a well-written paper, give you about 75% of the whole gist of the thing. And then try to read the discussion conclusion, because you want to understand kind of how they figure things out. So that's how to read a scientific paper. Now, if you're asked to present a scientific paper, usually that's going to be, at least at the undergraduate level, through PowerPoint. There are a number of components that I think are important for you to include. Now, my opinion is not universal. Other professors have specific ways they want you to present journal clubs. That's fine. This is sort of my interpretation. But of course, I think my interpretation is best, so that's what I'm going to teach you. And this is how I'm going to do it in the next few slides. So the first thing we're going to do is put up a title slide. This has the titles. For the title of the paper, the authors, the institution that the author works for, and the journal that it's published in, and specifically the date, right? You want people to know that the year is most important, right? You want people to know, is this a 2023 paper where, you know, it's probably got everything up to date? Or is this a paper from 1995 and maybe things have moved past it? Then I think one slide summarizing the key points of the paper. Right? So remember the old saw. If you're going to present something, you tell them what you're going to tell them, you tell them, and then you tell them what you told them. Right? You're repeating things 3 times. The first thing is the key points. You're already telling them this is what the author's main points are. So then you're going to start with the introduction. You want some slides that introduce the topic to a naive audience, right? You're really presenting mostly to undergraduates who are not familiar with the area. If you're doing graduate journal club, then it may be a little bit different. And oftentimes, students aren't sure, you know, can I take something that's not from the paper I'm doing? And yes, you can, particularly for the introduction, right? There might be pictures or graphs that describe things much better than the paper itself, because the paper is geared towards people who are already experts in this field. So you can always borrow some stuff from Wikipedia or other articles or something, or Google or something like that. The methods, right? A couple slides explaining how the experiments were done. This is pretty straightforward. The results, I would use one PowerPoint slide per figure with a descriptive title. So not just figure 1, figure 2, table 1, right? Tell me what it's about. You don't necessarily have to include every single figure in your presentation. If, in your judgment, this figure is redundant or doesn't really contribute to the overall story, or maybe it's going to make my presentation way too long, then skip it. It's okay. Basically, just want to tell the basic fundamental story without kind of getting too distracted. The discussion conclusion, one slide restating the experimental findings. So the key points, you tell them what you're going to tell them. The introduction method results, you tell them. The discussion conclusion, you tell them what you told them. You're just restating the main points of the paper. Hopefully once you've said them 3 times, they will stick with people. And then I also like to add, this is not universal by any means, but I think this is important. A sort of a last slide identifying either potential problems with the paper and or speculating on what future experiments might be useful. Now the first thing, potential problems, is because not every paper, in fact most scientific papers, are not perfect. As you learn to read them, you'll learn to pick out some things that you think maybe the authors did less than 100% perfect. And sometimes the omissions or the mistakes are important. The more familiar you get with an area, the better you'll be able to do this. But sometimes it's so glaring that you're like, how did this thing get published? That's part of a journal club. That's part of what you are supposed to tell the audience. Is, I read this paper thoroughly and critically, right? And I was able to find some things that I think are problematic. And then the last thing you kind of do is want to leave people with, okay, this is where this paper kind of sits in the universe of scientific understanding, right? There was stuff that came before. This paper used that information to make a unique contribution to the science of study of lead poisoning, for example. And then here's some places they might want to go with this. Here's some next steps that might be useful. So kind of putting it in context of, you know, like I talked about, like how Darwin started something and then someone else took it along and then someone else took it along from there. Your paper is just part of a long continuum. There's a whole bunch of stuff leading up to it and there's going to be a whole bunch of stuff after it. So if you kind of put it in that context, I think it helps people grasp, you know, the significance of what you're talking about. So that would be, in my opinion, how to present a scientific paper. Like I said, other professors might have some things that just, I think most of them would agree pretty generally this is the right format. You know, some people have their little quirks, but this is obviously the best way to do it. All right, so now I'm going to do exactly what I told you I'm going to do. I'm going to present this paper like I was doing a journal club. So the paper I'm going to talk about is this one. Toward the 21st century, Lessons from Lead and Lessons Yet to Learn. It's by Ellen Page Silvergeld. She's a member of the Environmental Defense Fund and also the program in toxicology at the University of Maryland, Baltimore. And this was published in Environmental Health Perspectives in 1990. So this is kind of an old work. There's more stuff that we know.
Dad wrote this.
So here are the key points of Silvergeld's paper. I think there are 4 of them. So lead, right, the element lead is a known neurotoxin, especially for children. Silvergeld argues the effects of lead on children are, quote, without threshold. And what that means is there's basically no safe level of lead in children's bloodstreams. Right? Lead is damaging from the first atom, really. You can't say, as long as you're below this number, you're okay. So the effects are without, there is no threshold. Just like, right, we talk about action potential. If you don't get up to the threshold, it's just going to go back down. In the case of the effects of lead, there is no threshold. Once lead gets in there, it'll start doing damage right away. And the more lead you have, the more damage you're going to have. The way lead works as a neurotoxin, right, something that's toxic to nerve cells, brain cells, is that it affects neurotransmitter release at the synapse. Remember how we talked about different things affecting neurotransmitters at the synapse? Drugs of abuse, antidepressants, toxins also. A lot of them have their effects at the synapse. And Silvergeld concludes by saying the effects of lead are essentially irreversible. The damage to the nervous system, you really can't fix it once it happens. And so her argument is that it's much easier to prevent lead poisoning than it is to treat it once it's happened. Okay. So those are the 4 key points of this paper. So let me tell you a little bit about lead. So lead has a long history. Humans have been using lead for thousands of years. It is a highly malleable, abundant metal. It has a low melting point, which makes it very easy to use for craftsmen. And in the thousands of years ago, when you couldn't make extremely hot fires, lead was very convenient because it melted at a low melting temperature. So lead has been used for thousands of years by humans. Lead beads have been found in Turkey that date to 6400 BC. The Romans, right, were very productive, had productive lead industry, smelted over 80,000 tons of lead per year. So huge amounts of lead in industry, in human environments is not new. It goes back thousands of years. In modern products, lead is in a wide variety of modern products. Most batteries, right, the disposable kind are called lead acid batteries. They have lead in them. Solder, so if you're ever doing soldering for, say, pipes, including domestic plumbing pipes, a lot of solderers have lead, although a lot of those have been replaced. Paint, lead used to be in paint, although again, it's banned in the US since 1978. Lead used to be in gasoline. It's being phased out worldwide. It's been phased out in the United States for almost 50 years. I think most of the world has phased out leaded gasoline by now. But it was still a particular problem even in 1990 when Silvergeld wrote this paper. And some ceramics, some ceramic glazes, right? You can usually, you buy like a bowl or something at Target, and if you flip it over, it'll say something like, not for microwave use or not for food use. If it says not for food use, that usually means there's lead in the glaze. So just like lead has been known for thousands of years, the neurotoxicity effects of lead have been known for thousands of years. We know that lead, when it gets into the body, accumulates in the soft tissues, like muscles, nerves, and also in bone. There are descriptions of what looks like lead poisoning in literature from ancient Greece, Rome, and China. So it's been known for a long time that lead can make people sick. The risk to both adults and children. For adults, you often get headaches, irritability, anemia, right? Not loss of red blood cells, not a good supply of oxygen to your tissues. Children, however, are much more susceptible because they're developing. So lead poisoning is characterized by developmental delays. Children who have lead poisoning walk slower, learn to read slower. They show attention and behavior problems, lowered IQ, hearing loss, and slow growth. So lead is a significant toxin to children. All right, so like I said, since this is a review paper, there is no method section. Since this summarized primary research papers, so there's no real method section here. If this were a primary research paper, I'd have a list of exactly how they did the experiment, where they got their lead from, things like that. Okay, so here's Silbergeld's results. Results figure 1, right? Here's my descriptive title, Mechanism of Lead Toxicity. This is how lead damages neurons. So right here we have a synapse. This is a nice square synapse. This is the presynaptic terminal. So lead enters the presynaptic terminal, that is PB. It increases the release of intracellular calcium. So this is the sarcoplasmic reticulum, which is a storage place for calcium in the cell. When lead gets in, it causes the calcium to be released in large amounts. Calcium is an important ion for the release of neurotransmitter vesicles. So the more calcium you have, the more neurotransmitter vesicles are going to exocytose at the presynaptic cell membrane, and more neurotransmitters are going to be released into the synapse. Okay, and more is not always better. In this case, more can be problematic. If too much neurotransmitter is released in the synapse, the effects on the postsynaptic cell, which is in the picture here, can be severe, up to and including it's called excitotoxicity. Basically, if it's carried on long enough, it can kill the postsynaptic cell. And so since lead has such a strong effect on neurons, we know that there are toxic effects on early neural development, so particularly in infants and children. So here is table 1. This shows that lead accumulates in synapses. So this is an experiment that was done in vitro, so not in living things, but in culture dishes. And so basically what they did was they created artificially some synaptic membranes, synaptosomes, and what are called synaptosomal ghosts, which are empty. So these are actually made from actual synapses. So you take the neural cells, you culture them, and then you put them in a buffer where the cells break up, but the little end of the nerve terminal, the synapse itself, is left intact. So that's a synaptosome. The synaptic membranes are just the membranes of the synapse, nothing else. And then synaptosomal ghosts are these, but they're empty. They have nothing inside them. No calcium, no nothing. So the experimenters looked to see how much lead was bound, and they used a radioactive isotope of, so looking at calcium uptake. Okay. So basically they were looking at how much calcium was taken up, because remember the method was lead causes increased release of calcium, and they used different concentrations of lead. And so at 0 lead, so this much calcium is taken up by the synaptosomes, by the synapses. Very little is in the synaptic membranes or the ghosts. So most of it is taken up by the synapses. We can be pretty sure that is similar to what you'd see in real life. Then what they have here are increasing concentrations of lead, right? So 10 to the minus 8th is 10 times smaller, 10 times less than 10 to the minus 7th, which is 10 times smaller than 10 to the minus 6th, and so on. So lead concentrations increasing. And as it does, the amount of calcium the synaptosomes take up increases, right? Which is just like what the hypothesis was. The more lead, the more calcium is released, the more neurotransmitters released, and that's bad for the nervous system. The synaptic membranes and the synaptosomal ghost are basically controls, so they don't really show this increase, showing that it's specific to actual nerve terminals. And the lead is not in the membrane, or the calcium is not in the membrane, it's somewhere inside the synaptosome, probably inside the sarcoplasmic reticulum, which is part of the calcium storage of the cell. So this shows you that with more lead, you get more calcium release. Okay, so this is the second figure that she presents. This is the effects of chelation of lead in children. And so unlike the previous figure, this was actually a clinical study done on children, patients who had suffered lead poisoning. So chelation is a treatment where somebody has been exposed to heavy metals, including lead. There are drugs that chelate the heavy metal, so they isolate it from the blood so that it gets excreted. And so chelation treatment for people who suffered lead poisoning, who show high levels of lead in their blood, does lower the blood lead levels in exposed individuals. So, and this shows you, this is a study of children who were who underwent chelation therapy. So this is the lead level in their blood. So this is the lead level in their blood at initial diagnosis, and then follow-up after chelation treatment. So this was an experiment that showed that chelation treatment reduced the amount of lead in the blood, right? Before chelation, the subjects showed between 50 and 80 micrograms per deciliter of blood. After chelation, it was reduced to somewhere between 45 and 20 micrograms per deciliter of blood. So the chelation did work to reduce the amount of lead in the blood. However, the paper points out, this is not necessarily correlated with a reduction in neurological deficits. So even though these children were treated and their blood lead levels dropped, the damage may already have been done by the time you can diagnose lead poisoning, which again means that treatments are not necessarily the most effective, right? The damage is done already. You can lower the blood lead levels, but the brain cells, the brain has already been affected. So this is a rather complicated figure, and don't worry too much about all the details. It's a summary of the biological effects of lead physiologically, but what I want you to focus on are these two things circled in red. So, and just as an aside, oftentimes if you show a complex figure like this, you're probably not going to be going through every single bit of it, so it's good to kind of highlight the stuff we're going to talk about so people don't freak out and think they have to read the whole thing. So I just want you to see that one of the effects of lead, right, is reduced hemoproteins. Hemoproteins include hemoglobin, which is your oxygen-carrying protein in your blood, which is why you often see adults with anemia who suffer from lead poisoning. But the main worry here in this paper is this disturbed role of calcium. So it impairs calcium in neurotransmission. It's toxic to neurons. It also impairs calcium as a second messenger. When we talk about signal transduction, calcium is a very common second messenger. That also affects that function of calcium. So not just in presynaptic nerve cells, but in other cells, lead can be toxic. So these are just a couple of the effects of lead, and this one particularly is what Sylvia is really focusing on. Okay, so discussion conclusion slide, right? This is the same thing that I just talked about. This is the same thing that I talked about with the points. So lead is neurotoxic to children without threshold, right? There's no safe level of lead and is unlikely to be easily treatable. Therefore, prevention is preferable to treatment, right? Not getting the lead in the first place. The mechanisms of lead toxicity at the synapse are generally well understood, right? We know that it leads to release of excessive calcium, which leads to release of excessive neurotransmitter, which is toxic to the nerve cell. So this point was actually made in Silvergeld's discussion, but makes an interesting point. So, right, we talked about, or she talked about in the beginning, how lead accumulates in the soft tissue, but also in the bones, right? And stuff that accumulates in the bones is often present for a long time, sometimes your whole lifetime. One of the problems of lead in the bones is that women who are pregnant or postmenopausal, so after menopause, is their body starts to cannibalize their bones if they don't get enough calcium. And by going into the bones and removing calcium, that also liberates the lead that was isolated in the bones, so it now becomes part of the bloodstream. That's where it has its toxic effects. Now, the figure for this was not included in the review, although the reference was provided, but it's particularly interesting because of the fact that removing lead from a population may not mean that everybody is immediately safe. Right? There are people who have accumulated lead in their bones, children, teenagers, and so when these women get to childbearing age or older in 20, 30, 40 years, the lead that's accumulated in their bodies may end up getting released. And so they may suffer lead poisoning down the road, even though they're fine now, because the lead is sequestered away in their bones. Pregnant postmenopausal women too suffer from low blood calcium. Right, taking calcium supplements is a good way to keep your blood calcium up, so that your body doesn't start to leach calcium from your bones, because your body is going to try to keep your blood calcium at a steady state. If it has to take calcium from your bones to do it, it will, and that's going to make your bones fragile, which leads to osteoporosis in postmenopausal women. Also, in the case of people with lead poisoning, it remobilizes that lead that was kind of sequestered away inside the bones, and now it becomes a problem. So, problems, future directions. So, since it's a review paper, there's really no experimental design to criticize, and lead toxicity is not very controversial anyway. Lead toxicity, like I said, has been known for thousands of years, so there's not too much to criticize about this paper. A couple of future directions that I think might be helpful. I think further studies of adult women to establish the potential release of lead from internal tissues, not just bone, but other tissues. Particularly when adult women who have a chance to become pregnant, I think it's important to study how much lead is already circulating in their blood and how much can get mobilized during things like pregnancy or menopause. And also kind of to look at the effects of the reduction in lead usage, right? So, as I said, in the 70s, a lot of lead paint and lead gasoline was phased out in the United States. Lead acid batteries are still around, right? That's most of your Duracell and Eveready type batteries, although it's being replaced largely by the lithium-ion batteries. And so I think it would be important to study this reduction on the prevalence of lead poisoning. Again, since this paper was written in 1990, it's been 34 years. I'm sure there have been significant reductions in environmental lead. And hopefully, the situation is looking better now than it did in 1990. All right, so that is the Silvergeld paper. So that's fair game for the exam. I might ask you questions about this paper. I might ask you questions about my PowerPoint. So that's everything that's going to be on exam 1. Exam 1's going to be on Friday, right? It's going to be in class. right? Pretty typical format. You've got true, false, multiple choice, a couple short answers, and essay. The 2 essay questions are now live on Moodle, so you can read them and prepare your response ahead of time. You can also, remember, bring an 8.5 by 11 sheet of paper. You can do both sides. You can type it, you can write it, I don't care. Although, as I said, right, handwriting tends to be better for retaining the information than typing it out. So if you can't handwrite your cheat sheet, quote unquote, I think it's better for your understanding and your retention. And really, at the end of the day, that's what I care about. I don't care about your performance on the test. I care that you remember this stuff, or at least part of this stuff.